pela sua presença no nosso simpósio. Como não temos certeza de se temos hoje aqui na sala, e com certeza sim, no YouTube temos pessoas de fora do Brasil. Embora eu me sinto mais confortável em português, vou passar a minha fala para inglês só para apresentar ao senhor em nome da instituição, tá bom?
Tudo bem.
Então, dear Professor Marcelo, thank you very much to be with us today. In the name of our student and professor from the graduate program in biophysics and from the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro, it's a pleasure to have you with us today, and we want you to be at home in our symposium. And now I'm gonna pass the word to Professor Vanderlei de Souza, that is gonna say welcome to us in our presentation. Thank you very much.
Okay, thanks, Susana. It's a pleasure for me to start this second week of conferences that are part of a symposium on cell biology, microscopy, and parasitology that is also a discipline for the graduated students of the Biophysics Institute and other institutions that are part of the complex of graduate courses in Rio de Janeiro. And today we have the privilege to have here one distinguished Brazilian scientist, Marcelo Jacob-Lorena, that has a long career in the United States with important contribution to these fields. And we will hear more about who is Marcelo Jacob-Lorena by our colleague, Professor Fábio Mendonça Gomes. Fábio is with you now.
Okay, hello all. So Marcelo Jacob-Lorena was born in São Paulo, so he's a Brazilian, as Professor Vanderlei was saying, and he did his undergraduate studies at USP, at University of São Paulo. After that, he went to Japan for his master's thesis, and later he did his PhD studies at MIT, at the United States, where he has been there since that time. He began his first position as an assistant professor in Case Western Reserve University, and later he moved to Johns Hopkins, one of the most important schools of public health in the world, where he currently holds a position. He started his career working with gene expression on Drosophila, and later he moved to work with mosquito transgenesis. His lab was the first to produce a genetically modified mosquito that is refractory to Plasmodium, and his research has focused on the life cycle of the malaria parasites inside the mosquitoes and also in the mammalian host. At present, his lab is exploring alternative strategies by engineering bacteria that live in the mosquito gut and will produce anti-malaria compounds. And also, more recently, he also started working with Plasmodium biology on the mammalian host, including mechanisms of sporozoite liver invasion. He has over 160 publications in peer-reviewed journals, such as Science and PNAS. He has chaired for 6 years at the World Health Organization Committee on Molecular Entomology and is on the editorial board of several scientific journals. In 2009, he was elected Fellow of the American Association for Advancement of Science, and in 2016, he was elected Fellow of the American Academy of Microbiology. I would also like to point that Marcelo is amongst the nicest person I have had the pleasure to work with, and an amazing person to discuss science. With that said, I would like to thank Professor Marcelo Jacob-Lorena for being here with us and for taking the time to present his work. Please, Marcelo, whenever you're ready, you're free to go.
Thank you. Let me. Can you see the screen?
Yes, you just have to swap the settings again on display settings.
How about?
And you're good to go.
Oh, good. First of all, I want to thank the organizers, Vanderlei, and the organizers for making me part of this wonderful series congratulations, Wanderlei, for organizing all of this. And I feel very honored to be part of it. And it's nice also because I feel very close to my roots, Brazilian roots, and it's nice to be at least virtually present in Brazil. So today I'm going to present, let's see, is this a story that started many, many years ago? And the main players are Sibao Wang, who was a postdoc in my laboratory when that started, Abdoulaye Diabate, who is a head of a research group in Burkina Faso. And Diabate is a world expert on swarming, mosquito swarming. And then Joel Vega, who was also a postdoc in my laboratory. And both Joel and Sibao moved on to have their own groups, and I will mention them again at the end of the talk. So again, this is a story that is going on for quite a while. And first of all, I'd like to introduce you to the malaria problem, which I think most of you are aware. It kills about 500,000 people per year, mostly African children in Africa. And so it is a very serious disease. And one very unique feature of malaria compared to other infectious diseases like AIDS and tuberculosis, malaria needs the vector mosquito in order for transmission to occur. AIDS, tuberculosis, that also kills a very large number of people, they are transmitted directly from person to person. So this is the cycle. And then if you eliminate the mosquito, you don't have transmission. And this has been taken advantage of. And the main tool that we have to fight the mosquito has been insecticides that kill the mosquito. So some statistics, World Malaria Report from WHO, there are two facts here. Between 2010 and 2018, there was a very large decrease in the number of malaria cases and deaths. And that is mostly because of the distribution of insecticides impregnated bed nets. However, from 2014 to 2018, there was no change, no decrease anymore. The decrease stopped and it stayed at similar levels. Why is that? It's mainly because the mosquito became resistant to insecticides. So the insecticides on the bed nets didn't kill the mosquitoes anymore. The mosquitoes changed behavior. They started biting people outside the houses instead of inside the houses. And it is a conundrum. It is a very big problem looking forward. So what this all means is there's a critical need to innovate and find new ways to target new targets to control the disease. And so that's part of what's in the back of our minds when we do research. And as I just mentioned, mosquito is critical in the transmission of the disease, and we have to know the mosquito better. So one question is, in order to reproduce, Anopheles mate. So the male and females have to mate. And how does this happen? It happens in swarms. So these are swarms of male mosquitoes only, and it happens at the end of the day. And what happens is that male mosquito swarms, and then female mosquitoes come individually into the swarms, and that's when the mating occurs and reproduction. So, and the mosquito that rests, the mosquito that's not mating, where is that male mosquito? Very frequently, it's resting inside the houses. So this is the situation. So we have mosquitoes that are at the end of the day are swarming, and others that are not. And the simple question that we ask, just a curiosity, is what are genes involved in the induction of swarming behavior? So are there any genes that are different in mosquitoes that are swarming compared to mosquitoes that are resting in the houses? That was basically the question that we asked with this project. And what we argued is that the genes involving influencing behavior are most likely to operate in the brain. And for this reason, what we did is then collect male mosquitoes from swarms and from houses at the same time of the day. So we have the male mosquitoes from swarms and houses that we can compare. And then this was done by Diabate. He sent us the mosquitoes, and we extracted RNA from the heads of these male mosquitoes and compare the transcript representation using microarrays, compare swarming with non-swarming. And what we found is that a very small number of genes were differentially expressed between those two groups of mosquitoes. Here is the upregulated genes, these are the downregulated genes, and a total of about only 90 genes out of the 15,000, that means 0.6%, less than 1% of the genes are differentially regulated between resting and swarming male mosquitoes. So this is, in a way, it's a good start for this project, because we have relatively small number of genes that are differentially regulated. So what are those genes? So we have, these are functional groups, immunity, metabolism, clock, cytoskeletal, structural, cell division, and so on. And interesting, most of the immune genes were downregulated in swarming mosquitoes. And perhaps not surprising, most of the metabolism genes are upregulated in the swarming mosquitoes. They're flying and they're using energy. But what caught our attention is that all the few clock genes that were differentially regulated were upregulated. All of them were upregulated in swarming mosquitoes. This is clock genes are the ones that regulates the circadian rhythm between day and night. So that was the clock genes are where we focused on. And another test that we did after we got those results is to see if the comparison by microarray also was true when we did quantitative RT-PCR. And for each of the genes that we tested, there was we confirmed the differential regulation of those genes into so these are true results. The microarray results were confirmed by quantitative RT-PCR. And as I mentioned, the clock genes were what caught our attention. And also, what caught an attention is a gene called desaturase 1. And I will come back to this gene, and I will tell you why we became interested in this gene. But let's start with the clock genes. So, does the clock gene has anything to do with mating behavior? So, what we use is double-stranded RNA. We inject double-stranded RNA with sequences to key to the genes that we want to inhibit the expression. And GFP is green fluorescent protein was used as a negative control because GFP is not naturally expressed in the mosquito. And we use the two key clock genes, timeless and period. We inhibit them. And if we inject double-stranded GFP, we have a certain level, about 65% of mated mosquitoes. But when we inhibit either timeless or period expression, the number of mated mosquito decrease significantly. And this is true for Anopheles stevensi, which is an Asian mosquito, and Anopheles gambiae, which is the main African vector of malaria. And in both cases, mating is reduced when we inhibit the clock genes. This was done in the laboratory. We then asked if this also is the case under more natural conditions, and we use what we call mosquitosphere, which is an enclosed space with vegetation and chambers in it where we can release mosquitoes and test them for all sorts of parameters, including behavior. So when we use the semi-field conditions and when we inhibit timeless period, we saw that the swarming, so this was a very big experiment done of Joel together with Diabate. He went to Burkina Faso to these experiments. He injected double-stranded RNA in hundreds of male mosquitoes and then released them into these chambers and studied then the swarming. Swarming heights of the mosquitoes was decreased when timeless or period is inhibited, and the number of male that swarm was also significantly decreased, and insemination rates, the number of females that had mated with the male mosquitoes also decreased. So even under natural conditions, swarming and mating is dependent or regulated by the clock genes. We also looked at the effect of temperature in mating activity. And this is the normal temperature is 27 degrees. We tested low temperature, 19 degrees, high temperature, 34 degrees. And when at normal temperature, about 60% of the mosquitoes mated. At lower temperature, 19, very few mated. And when it's above the normal temperature, the number of mated females also decreased. And we use these mosquitoes to look at the period, expression of period. And here is light. These are mosquitoes kept under light and dark conditions, 12 hours of light, 12 hours of dark. And this, in blue here, is normal expression of period. If you go to 19 degrees, the pattern of expression is altered. It decreases much faster, and at high temperature, also expression of period is lower than normal. So there is a connection between temperature, mating, and expression of clock genes. We also looked at the effect of light. So the normal conditions are light, dark, 12 hours of light, 12 hours of dark. And under these conditions, we have more than 70%, above 70% females mating. But if we entrain the mosquitoes light, dark, and then switch to light, light, never make dark, that inhibits significantly the mating of the mosquitoes. And this is matched by expression of the period gene. So the maintaining light, light, the expression of period gene is significantly decreased. So the conclusions of what we saw so far is that clock genes regulated swarming and mating, and light and temperature affect mating, including expression of the clock genes. So this was interesting, but when we looked at the list of differentially expressed genes, we noticed this desaturate gene was highly differentially expressed between swarming and non-swarming. And then we went to the literature and we saw that previous studies in Drosophila has linked this gene with behavior, neural and non-neural expression, and the emission of sex pheromones. Another paper looked at it and also this to sex pheromones. So we then were curious to find out, does this desaturase also play a role in the behavior of anopheline mosquitoes? And here is, we saw that that there is regulated between swarming and not swarming. Here we saw regulation as a function of circadian regulation. Light, 12 hours, 12 hours of dark. And it goes up and then goes down. But if you inhibit timeless expression interfere with the circadian gene expression, the circadian expression of desaturase goes away. It's disturbed. And the same thing for period expression. When here's desaturase normally, and if you inhibit period expression, it's the rhythmic expression between day and night is affected. And if you also alter the light, instead of change from light dark to light, the expression is of desaturase is also decreased. So there is a intimate connection between clock genes and the circadian rhythm and desaturase expression. We also looked at the behavior of movement. This experiment was done where you put individual males in little tubes and then keep those tubes in light and dark conditions. And in each tube, there was a laser beam. And every time that laser beam is broken by the movement, if the mosquito moves in that tube and breaks the beam, then the apparatus registers that. So during the day, during light, there was little activity, and then when it becomes dark, the activity increases tremendously, and that is what normally happens. And then it goes down at the end of the dark period. But if you inhibit the desaturase gene, this dramatically affects the behavior of the mosquito. It moves much less. So there's a connection between desaturase and mosquito behavior. We also looked at mating. If you inhibit desaturase gene expression, mating goes down, both for the Asian vector and for the African vector. Inhibition of desaturase inhibits mating. So we then looked at the cuticular hydrocarbon of male mosquitoes. So just do mass spectroscopy, and we saw quite a large number of peaks. This is from a normal male cuticle. But when we inhibit desaturase, there were two main changes. This peak here of tricosane was significantly lower when the desaturase is inhibited, and so was the heptacosane, the one with the 7 carbons was also significantly lower. So we then, based on this result, we tested, does this tricosane or heptacosane have a direct effect on mating of the mosquitoes? So what we did is with perfuming, we spray the mosquitoes with either the tricosane or heptacosane, and then looked at mating. This is a control. Here we have 50% insemination rate. If we perfume or spray tricosane on the mosquito, there was no significant difference. But when we spray the mosquitoes with heptacosane, there was quite a very significant increase in mating. So heptacosane and not tricosane have effect on mating. And remember, heptacosane synthesis is under control by desaturase 1. So the heptacosane is a mosquito pheromone candidate. So that's the first one for mosquitoes. So that's how we got from swarming, looking at swarming and non-swarming mosquitoes. We found 90 genes differentially expressed. We found that circadian rhythm were differentially expressed between swarming and non-swarming. And we found that if we inhibit those, swarming and mating are both in the lab and semi-field conditions are affected. And desaturase is regulated also by the circadian genes and promotes the synthesis of heptacosane, which is the mating of pheromone. So that's basically our finding. We went from a simple question of comparing swarming and not swarming mosquitoes, and we get to circadian rhythm and pheromone. So the credit is Sibao Wang and his laboratory. He now is a head of a large lab in Shanghai of the Chinese Academy of Science, and his members of his group contributed significantly to this work. Joel moved from my lab to the NIH and has a big lab and quite successful lab right now. And Diabate is, as I mentioned, is an expert in swarming, but not only that, is one of the African leaders of vector biology and works in Burkina Faso. And financial support by NIH, Bloomberg Finance, Philanthropy, and the Chinese Academy of Science. Thank you.
Hello, Marcelo.
Yes. Yes.
Okay. Hey, beautiful work. It's very interesting where this thing is leading you right now. So we have a few questions arriving. I will start organizing them, but just for your information, we had more than 100 people watching you on Google Meet and also on YouTube, if we put them all together. And I'm sure from our experience with the previous presentations that a couple hundred more will be watching this video over the next couple of days and week. So pretty good. So let me see. Can I start asking you if you could talk a little more about these clock genes? So do you have an idea of exactly what they are and what are the molecular mechanisms that they are using to regulate, for example, the desaturase expression?
Yeah, the clock genes are very, very well studied set of genes in Drosophila, and one recent Nobel Prize was dedicated to the discovery and the study of the clock genes. And Mike Rosbash, who was a former colleague of mine at MIT, was one of the persons who got the Nobel Prize for the study. So it's a very well characterized group of genes, and we found that it is regulated, the desaturase genes is regulated by the clock genes, but we don't know how this happens. This is quite, as you probably notice, is unpublished work is quite recently that it came to the stage, and we haven't had a chance to make a molecular bridge between desaturases and clock genes.
Yes. Yeah, thank you for being generous and presenting this data. So I remember in the past you have used promote, and like you have been able to identify promoters like related to early digestion and things like that to do transgenics. Do you think that you could also identify promoters that are related to the circadian rhythm, and would they be useful for similar approaches? I'm not sure if I'm going very far right now, but I was just wondering after what you mentioned.
Let me see if I understood correctly the question. If we could identify promoters that are regulated by the circadian rhythm, is that the question?
Yes. And
yeah.
Would they be interesting for transgenics and
yeah.
things like that?
Yeah, that's a good point. I hadn't thought about that. That is perfectly possible. And one would need to make the transgenic mosquito linked, but this is perfectly possible. And again, is one direction this can take. And the other direction is obviously if we identify if this is can get confirmed as a pheromone, this also has practical implications in the control of the mosquito.
So have you actually tried to use the heptacosine as a mosquito attractant?
No, we haven't. We have just studied this, the mating behavior, but not. We don't know if it would attract, for instance, females specifically.
I see. So we have a few questions coming from YouTube. So Celie Dapres, you might know her, I guess.
Yeah, I know her well.
Yeah, so she was asking if there is data in the literature showing how clock genes affect plasmodium infection in both the host and the vector.
There are some recent studies that look at periodicity of plasmodium in the reproduction in the mammalian host, and this is something very recent only, and it's some people connect the periodicity of that effect that plasmodium reproduction in the mammalian host is periodic has been known forever, but now some people claim that this is connected to the host circadian rhythm, and others say that it's endogenous by the plasmodium. So it's a little bit of a controversial subject right now.
And we have two questions from Alison Julio and Rafaela Bruno. They go pretty much into the same direction. So do you know if this set regulates female pheromone, or is it restricted to male mosquitoes only? And were you working with, did you try to work with both male and female mosquitoes, and did you see any difference?
Yeah, that's a good question. As far as I know, we have not tested the expression of desaturase in the female. That's a very good question, and that's something that we should probably consider doing. I will make even a note of. Yeah, that's a good question, and the answer, unfortunately, is I don't know if it also operates in the female. I would guess that it's male specific, but we don't know if that's the case or not.
Ideas for the future, I guess. So Pedro Oliveira was asking you if you could give more details on the metabolism genes that are modulated by swarming, and how much, and what about other lipid metabolism genes in addition to desaturase?
Oh yeah, there are quite a few other genes, but we have a list of them, but we have not done any experiments with them. So it's something we have a relatively small lab, and we cannot pursue everything. But we are writing this work up now, and we'll be obviously very glad to share the lists for other people who would like to pursue this line of research.
Okay, that's great. Thaisa Villa was, so you're showing some data on temperature effects. So she was asking if you think that global warming could affect mating enough to exert a natural control over malaria infection?
Yeah, so what I showed is the effect of temperature on mating. So one could think of the global warming affecting the proliferation of the mosquitoes, and it is possible that it will affect the number of mosquitoes. But then what probably will happen is that it will shift to places which are now at lower temperatures that will warm up, and so the mosquitoes will just move away from certain areas and move into other areas.
I see. Do you know if anyone started to see anything like that?
If anybody's studying this?
No, if anyone started to actually notice some kind of movements like that in the, I don't know, past few years?
Yeah, I don't know for sure, no.
Okay. So, Ana Bahia, Ana, do you want to address the question yourself? I'm not sure if you're around.
Okay. Can you see me? Okay, thank you, Marcelo, for the great talk. Do you think that the eptacosan can be used in traps aiming to the reduction of mosquito population in areas of high malaria transmission?
I
do you think that it could be used as a control?
I think so, because that would disturb the mating behavior of and possibly attract females. So I think this is it's a possibility. It's a real possibility, but more work needs to be done to prove the practicality of this.
Okay, I see. Thank you.
Thank you, Ana. I'm not sure if anyone else has a question. I have another question, if I have the opportunity. So, Marcelo, I know it's not exactly what you are discussing right now, but you are one of the leading areas in the world. So, could you talk a little more? And we have people from graduate programs and things like that. So it would be interesting if you could give them your opinion, or at least your view on that subject, about the roadmap to deploying transgenic mosquitoes in the field.
Yeah. Oh, this is an interesting topic. We could spend another hour just talking about that. But anyway, first of all, there is a consensus, a very clear consensus, that just producing a lot of transgenic mosquitoes and liberating them into the field would not work. So you need what is called the gene drive, a way that make the gene of interest, for instance, the gene that inhibits plasmodium development, into the mosquito to spread into the population. Another, this is one way to use gene drive. Another way to use gene drive is to inhibit reproduction. So any mosquitoes that inherit a given set of genes does not survive and the population crashes. So these are two very different strategies. I am very much biased towards the population replacement or modification, meaning spreading inhibitory genes into the population, but this is not yet feasible now. On the other hand, the population reduction, so killing the mosquito, using gene drive to eliminate mosquitoes, that is in an advanced stage, and it will start being used in the near future in pilot studies in Africa. So this is getting close to reality. And so I'm, as I said, I think we should not go against any new approaches. I think this will be certainly helpful to kill the mosquito, and it should be tried. But the problem when you kill mosquitoes is if you have a lot of mosquitoes in a certain area, that means that biological niche, the conditions are very favorable for mosquitoes to live and thrive. And if you artificially kill these mosquitoes, you will leave an empty niche. So you'll get an area of that has very few mosquitoes, but they're very favorable. So as soon as you relieve the pressure, you stop killing the mosquitoes, it comes back at a very rapid rate, and that is the weak point of this. And it can either be if you stop, or if mosquitoes resistant to the killing show up. For instance, a mosquito from a different species of the same genus starts populating, and you have to switch the thing. So this is a long answer. I am positive about any new measures. I think that, and I think what will happen next with transgenic mosquitoes is implementation of gene drive to kill the mosquitoes. But I think that research needs to continue to improve this approach. Ideally, having every mosquito living and happy, but unable to transmit the parasite would be the ideal situation, and there would be very little pressure against this mosquito.
Okay, thank you very much for your perspective, and I hope we can move forward with like the authorization to do these trials in the field.
I'm just completing the thought. The alternative to transgenic mosquitoes is to engineer bacteria that live in the mosquito naturally to secrete anti-parasite molecules, and that's called paratransgenesis, and it is an active subject of our research, which I think has a lot of promise in my view.
That's for sure. Okay, so I have one last question that I missed when I was going through the list of questions, and then I will ask Professor Vanderlei de Souza to wrap up this presentation. So Rafaela Bruno was also asking if you notice any difference in the time the swarm happening after you did the silencing experiments?
Could you repeat that?
Sure. So when you did after you did the silencing of the clock genes, Rafaela Bruno was asking if you noticed any difference in the timing the swarm was happening.
This was not done because the way it was done is injecting double-stranded RNA to timeless or period, and then at the end of the day, just release them into the mosquito house. And so we could not, this is a huge amount of work to inject double-stranded RNA in sufficient numbers that you can see the swarming. And we could not do experiments about the timing. So we don't know.
Okay, great. Thank you. So I'm not sure if, because I know Susanna could not be, well, maybe she was not around by the end of the presentation. Susanna, are you there? Okay, so I'm not sure if Susanna is still around, but I would like to thank you very much in the name of our graduate school. We have a lot of students here. I'm sure that it was very important for them to have the experience of being able to talk to you and address your questions and also to see your presentation. And then I would like to ask Professor Wanderlei de Souza to make some final comments and wrap up for today's presentation.
Okay, Fabio, are you hear me?
Yes.
Okay, yeah, I would like to thank you again, Marcelo, for this wonderful talk. And I would like to make some comments in Portuguese for our students. Creio que a conferência do Marcelo mostrou a importância de aprofundarmos aqui no Brasil os estudos sobre esses agentes transmissores de várias doenças, tendo uma forte entomologia molecular. Quer dizer, as várias alternativas de interferência gênica no mosquito para interferir com a capacidade de que o ciclo do plasmódio, ou o ciclo da leishmania, ou o ciclo do Trypanosoma cruzi ocorra nos insetos transmissores, aponta para uma área que precisa ser mais estimulada e precisa ser mais incentivada no país. E nesse sentido, Marcelo, eu creio que seria muito importante podermos contar aí nos próximos anos com a tua presença física mais frequente aqui no Brasil, né? O Fábio já deve ter te falado das nossas iniciativas iniciais na Amazônia, no qual a gente espera poder desenvolver estudos mais aprofundados na área da entomologia, mas é preciso que os estudantes, né, de iniciação científica, mestrado e doutorado, que tiveram a oportunidade de assistir esta palestra, possam também se motivar a se engajar em projetos desse tipo. Creio que essa palestra foi muito boa, além dos dados científicos em si, mas no sentido de estimular os jovens e mostrar um campo aberto e relativamente pouco explorado no país. Eu, às vezes, me pergunto, se a gente elimina esses mosquitos, outros mosquitos virão, pode ser até piores do que os que nós temos, até mais perigosos. Então, é preciso alternativas de que a gente conviva com os mosquitos, mas que esses mosquitos não possam nos transmitir essas doenças. Acho que essa é uma abordagem ecológica mais interessante. Então, agradeço a participação de todos, convidando para a palestra de amanhã do nosso colega de São Paulo, da Escola Paulista de Medicina, professor Sérgio Sheikman. Obrigado a todos.
Obrigado. Foi um prazer e uma honra de participar.
Ok.
Marcelo, mais uma vez, obrigado, hein?
Ah, que isso.
Vamos manter contato e, por favor, fiquem seguros aí. A situação de vocês, eu acho que já está melhor do que a nossa situação aqui no Brasil, ao menos aí na região de Baltimore.
Tá, tchau. Obrigado a todos.
Abraço, tchau, tchau.